Well, thanks again, Maureen, for the introduction, and thanks again for the invitation. You do today, as Maureen mentioned, is really giving an update on this aptamer we've been interested in for a couple of years now, which targets von Willebrand factor we call DTRI-031, and looking at it in increasingly more sort of clinically relevant models, and show you some unpublished data now that we've been looking at it in really a large animal model, a canine model of large vessel occlusion, where we occlude the vessel for 6 hours and then try to treat it to open it back up. In addition, I'll show you some of the IND enabling, or the summary of our IND enabling studies that's helping us translate this into humans. And the important thing on this first slide is just to point out that this is a close collaboration between our group and the NIMG laboratory and the Department of Neurosurgery at Ohio State. As far as disclosures, co-founder of a company called Basking Bio, which has licensed patents from Duke on sort of the aptamer compositions that are used to help translate it and hopefully commercialize the approach. So today, really, I'm going to first do is quickly show you a few slides, try to convince you, although I think everybody knows that there's a major unmet need for treating particularly acute ischemic stroke. So ischemic stroke, if you have a blood clot and a vessel in the brain that blocks adequate blood flow to the brain and can compromise different parts of it. I'll show you a brief background of this aptamer. We published it before, and I think I even talked a little bit about it last year as part of one of the parts of the presentation. But mainly I'm going to spend time on the last two bullets about how we're evaluating it, and again, hopefully more clinically relevant models in canines, and then give you an overview summary of the results from our IND enabling PK tox work.
So the slides aren't switching, I'm not sure.
They aren't switching?
Yeah.
Good. Wonder why they are not switching. They switched when we...
They switched before, yeah.
What is happening? Why is it?
Okay, so now it's on the disclosure slide.
Or is it switching now?
Yes.
All right.
Perfect.
Technology. Anyway, so these are the things I mentioned in the overview. Not going to re-say it, but tell me again, Maureen, if it stops. I don't know why it stopped, but hopefully it won't do it again. So again, we got interested in ischemic stroke because unfortunately it's common and deadly and disabling. The only treatments that have been shown to improve stroke is to try to, as rapidly as possible, restore blood flow to the brain. All the neuroprotective strategies post-stroke, unfortunately, have not been successful. Our batting average from a therapeutic space there is 0, unfortunately. So really the focus, the only thing that's been shown to be valuable is to open up the circulation back to the brain. Again, stroke is common, second leading cause of death and disability. Unfortunately, 1.4 million people a year in the US, EU, and Japan experience strokes, even higher in China, unfortunately. It's growing, and 1 in 5 of us in the audience will, at least currently, if we're on the same trajectory, now through our lifespans, will experience an acute ischemic stroke where we have a blood clot in the brain. The outcomes of these tend to be poor, and it's actually the leading cause of long-term disability, and it's estimated to be about $230 billion in cost to the US healthcare system by 2030. So major problem. What's somewhat surprising to me, at least, was when we began this, is that very few people even get treated when they have stroke. So only about 10% of patients get the one approved therapeutic, which is TPA, targets fibrin, and that's largely because of bleeding risk associated with it. If you bleed, convert acute ischemic stroke into hemorrhagic stroke, it's about 40 to 50% mortality. So physicians are really scared of using TPA, and that means it has a very short and small therapeutic window. That's why you always hear you need to get to the hospital fast in 3 to 4 hours post stroke symptoms. More recently, it's actually a surgical procedure where in certain large vessel occlusions where you have a clot in a major vessel in the brain, people can do thrombectomy, and literally they go up into the brain with a catheter, put a cage basically around the clot, or put something through the clot and physically remove it. And so that's becoming more common, but unfortunately it's very kind of rare. You have to have the right clot, I mean, the class being the right vessel to have access, and two, you had to be able to have access to these very specialized facilities and trained individuals to even, you know, have access to this. So it turns out only about 15 to 20% of people get any treatment who have acute ischemic stroke. So there's, again, a major unmet need. Let's see if it goes. We got particularly interested in stroke partially because when people were pulling these clots out of brains, they started to look at sort of the clot structure. And what was interesting is, so people, when they were doing thrombectomy, got particularly interested in, as shown here on the right-hand part of the slide, is why TPA was often failing, particularly in large vessel occlusions. So these are, you know, they pull these clots out even after they've given TPA and it didn't help at all. And so when they did histopathology and immune staining and scanning EM of these clots that they removed, they sort of determined that the clots actually had sort of the structure is sort of highlighted in the left part of the slide in this cartoon. And so what they saw at the base of the clot was sort of a fibrin cloud. And this is what TPA is going after. And it's basically the bandage, or, you know, I like to say it's the patch on a tire when you have basically a flat tire that you patch. So you're blocking blood from escaping the vasculature or air out of the tire. But when you look at the rest of the clot, that becomes occlusive. So this is the base of the clot. The clot is propagating across the vessel, and ultimately you block blood flow, and that's the problem. What they saw is there wasn't so much fibrin there, but you saw densely packed platelets sort of in an inner core of the clot, and then that was surrounded by looser packed platelets that weren't activated as much. And as shown here, von Willebrand factor was found throughout this part of the so-called shell of the clot holding together these loosely packed platelets. So when we saw that, well, when they saw it, basically said part of the reason TPA doesn't work is TPA can't get through these barriers to even get to fibrin. I think the other thing that they saw is that, you know, you go and you dissolve this fibrin, and it's again like removing the patch on a leaky tire, and so that's why you tend to bleed. So we got interested because we've been targeting von Willebrand factor for a variety of reasons over the years. We thought in this case it might be good to actually try to target these clots and try to loosen them up in theory from the outside in versus going after something that was tough to target like fibrin. So that's why we got particularly interested in seeing if we could explore inhibiting VWF based on this clot structure information. So we had it after we actually optimized. I showed this before, this has been published. Again, it's fully modified. It's about 30 nucleotides, and then with the 5 nucleotide poly-U tail here that we put on here to make a antidote oligo or reversal oligo with. It binds moderately well, like 9 or 10 nanomolar dissociation constant, not picomolar, but low nanomolar dissociation constant. Again, we were able to make a reversible inhibitor to von Willebrand factor by simply scanning and making this what we call antidote oligo 25 here, which forms base pairs with this little tail and then unzips the rest of the aptamer. I'm not going to have time to show you today, but again, we've shown and published some of this, that this can be effective within a couple minutes in vivo in animals or even humans. And again, the clinical people like that because bleeding is such a concern in these patients. So we also published a couple of years ago mouse studies. So I'm going to quickly review them and move on to the dog work. So again, in mouse, what we did was to ask, could you open up an occluded carotid artery? Here we damaged the artery, waited 20 minutes or so, and then gave the drug, DTRI-31 at 0.5 mg per kg in this case, or TPA. You can see the drug, the Aptamer drug was more effective and opened up about 75% of the occluded vessels, whereas TPA was about 20 or 25%, which is similar again to what you see clinically. So that was encouraging. We went on and asked more about an embolic stroke type of model where you would make a clot in a test tube and then you would shoot it up into the middle cerebral artery of a mouse. And then you would ask, if you treated it, could you reduce stroke volume? And control animals where they either get a mutant aptamer or just PBS. This is the stroke volume. You can see that was significantly reduced when you again delivered the aptamer to these animals. If you do this with TPA, it turns out all the animals die because of hemorrhage. So that's why it's not shown on here. So this again was published, but we wanted to move to more clinically, relevant models. These are small vessels. Obviously, they're mice. They're not humans. It's not like when we're getting large clots in our brains. And also we wanted to look, this is only like 20 or 30 minutes. It's hard to keep mice down a long time and do these type of studies on anesthesia. So we wanted to move to larger animals and look at longer time. So we developed the canine large vessel occlusion ischemic stroke model. And basically what we did here is, again, take an autologous clot. So we draw blood from the dog, make it clot in a, a syringe, basically. We would image the circulation of the brain. So this is subtractive radiography, looking at circulation through the brain of the dog. We would inject the clot into the middle cerebral artery of the dog. And again, here it's we as obviously the neurosurgeons at Ohio State. I'm not going up into the brain of a dog with the catheter like they can. And what you can see is immediately, after this, when you look at by radiography, you're seeing loss of blood flow to say part of the brain, this hemisphere here. We would let the clot stabilize for 6 hours. And the reason why we did that is that's sort of 6 to 8 hours of when you usually see people show up post-stroke. Unfortunately, most people, again, don't make it into 3 to 4 hours into the hospital. And then we'd administer the drug or control. We didn't do TPA again because we were to kill all the dogs in our eye cup. People didn't like that. And then we'd simply monitor, did you open up the vessels by angiography, and then look at stroke volume by MRI at the end of the procedure. And the assays we used to look at this was we obviously looked where we're blocking platelet function. There's 2 ways we did that. It was a PFA 100, it's a kind of platelet function analyzer. It's a point of care platelet activity assay that people use in the clinic. There's also platelet aggregometry, which is sort of state of the art, after, it's not point of care, but I'm not going to have time to show you both. I'm just going to show you the PFA 100 data. It's similar to the aggregometry. And then we did, again, look for revascularization through angiography, and then we looked at infarct volume, again, by MRI in these dogs. And so what you see when you look at platelet function is essentially at this PFA 100 assay, the closing time of this assay, which is, again, a monitor of platelet function through VWF specifically, is that your closing time is usually 90 to 100 seconds. So these are dogs before they're treated almost immediately within 5 minutes at this dose of 0.5 mg per kg. The maximum time you can reach in this assay is 300 seconds. So you're essentially almost maxing out the assay, and inhibiting platelet function totally is measured in this approach. And you can see this gradually goes away over about 3 hours. I should have pointed out this drug is not formulated with the PEG or with the cholesterol or anything, because we wanted to have a short half-life. We wanted to go in, open up the vessel, and go away because they're so worried about bleeding sequela. So again, fairly short half-life, which is good in our case. And then we started monitoring, you'll see, at 1 hour post-treatment with MRI. So the most exciting data to me was when we started to see results like this when we did the angiography. So we treated 7 animals where you blocked them again, and then you gave them saline 6 hours post-treatment. You could see none of them opened back up. And so this is called a TICI score, which is thrombolysis and cerebral infarction score. And the TICI score of 0 means no reopening. And then we treated 8 animals with, again, the Aptamer 0.5 mg per kg, bolus administration 6 hours post. In 5 of the 8, we saw significant revascularization. And so 3 of them, we didn't see much. So again, the TICI score is 0, but a TICI score of 2A or B means that you get revascularization, partial revascularization to the infarcted area of the brain. It just means the filling rate is often somewhat slower. And then a TICI score 3 is normal. So that's just what you would see in a non-occluded. So there was 1 of the 8 animals, and then 4 of the guys that were partially. And so this is what you could see in these animals, that suddenly you would go from this to this. And when you talk to the neurosurgeon, he says that's like the difference between somebody walking out of the hospital and being crippled or not making it. So again, when they go and remove clots like this, again, and this is a large vessel in the MCAO, if you get this, that's what you want to tell a patient, they tell you. So that was encouraging, that 5 of these 8 animals. What about infarct size? And so it's interesting, if you look at all 8 of the animals that received the drug, there's not a significant reduction in infarct volume, but remember there's 3 of the guys that we saw no recanalization, and they're all similar to the negative control. So if you focus only on the 5 where we did see recanalization, then you see a significant reduction, not quite half, but 40% or so reduction in the infarct size, which is statistically significant. So that was encouraging to us as well. So with that data, we got excited from an efficacy perspective, and then we started to complete our IND enabling PK tox studies to hopefully move this into humans. I'm just going to quickly give you an overview of what we saw there, just to show you some of the tox studies, the safety farm and the genetic toxicity studies, because I thought it might be interesting for this audience, for the type of things that you have to do. The FDA let us do our rodent model in mice, and then our large animal and dog if you do 2 species. And again, the dose equivalents, remind people, we were intended dose in humans of 1 mg per kg. Our dog studies were 0.5 mg per kg. So we were just saying our intended dose was 1 mg per kg. They pointed out that the equivalent dose in a mouse is actually 12.3 mg per kg, and a dog is 1.8 mg per kg. And so they also wanted us to do up to a 50-fold safety margin over our intended dose. So we did a 1x, 10x, and 50x. So at the highest doses you'll see, we were treating mice with 615 mg per kg and dogs with 90 mg per kg of this compound. Again, bolus single administration. And the good news is that, as I'll quickly show you, is that all the tox information is very clean. We saw no issues. Here's the GLP mouse tox studies, again, going up to even 615 mg per kg in over 30 animals in toxicity and toxicokinetics. The data all came back where it was only mild, very mild findings, like, see temporary, piloerection, which is the hair standing up on the mice temporarily at the infusion site, et cetera. So thus was, from the CRO we worked with on this, who was Covance, they basically reported that there was no adverse effect levels up to the 615 mg per kg, and that's what we reported to the FDA. In the mice, same thing in the dogs, again, fewer animals, but again, 12 animals going all the way up to 90 mg per kg. No observed adverse levels up to at least 90 mg per kg. So at a 50-fold level above our intended dose, we were seeing good toxicity profiles. I'm not showing the toxicokinetics, but again, similar to what I was showing you in the PFA assay, depending on the dose, you can have a slightly slower clearance rate, probably because you're saturating the clearance pathways. But in general, by a few hours, you're regaining normal platelet function. In the safety farm studies, again, we did the 4 standard analyses that the FDA wanted us to do. I'm not going to show you all because of time, but again, all of them were very clean. I'll just show you the cardiovascular safety study in non-naive conscious dogs. Again, these are 3 and 3 animals, up to the highest dose of 90 mg per kg. And again, the results were there was no effects on mortality, any clinical observation, body weight, body temperature, blah, 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 ECG measurements in these dogs following even these high doses of the Aptamer drug. Similarly, in the genetic toxicity studies, everything was very clean, even at very high doses in the 3 standard assays. So all of this let us go to the FDA and submit for first-in-human studies, and we were permitted to do that. And that trial's underway right now, and it's almost finished enrollment. So in summary, what I tried to show you today about sort of looking at this compound and this large vessel occlusion more relevant model and the PK tox studies, would show that this Aptamer 0.5 mg per kg in a canine model where we stabilized a clot for 6 hours in a large vessel MCA resulted in revascularization in at least partial to full vascularization in at least 5 to 8 animals, and very significant where we clinically very valuable in half the animals in these dogs. And again, we didn't see any revascularization in the 7 animals that weren't treated with Aptamer. Probably not surprising because you let this clot, it was a big clot, we let it stabilize a long time. It also resulted in, if you did recanalize, it resulted in reduced infarct volume, not surprising because you're opening up the vessel, similar to what you do in thrombectomy. We didn't see any evidence of intracranial hemorrhage, but we still have the reversal agent in case that happens. Remember, these are healthy, normal animals. When you go and do this in patients, it's not going to be healthy, normal animals. So having this is still going to be really valuable if you don't see it in If you talk to clinicians, they love having this because they don't want to operate or have bleeding in these patients. Again, from a toxin safety farm and genetic tox studies, we had a very clean profile, no adverse events, even going up to very high doses in mice, in dogs. And that again let us go and get FDA approval for the phase 1 safety study, which is in progress, almost completed enrollment, and we'll have the results expected by the end of this quarter. And so hopefully next year I can show you those results and show how we're moving on to phase 2 proof of concept, actually in stroke patients. And with that, I'd be glad to take any questions.